How many times have you been told to take antibiotics for a bacterial infection? For example, let's say for a simple staph infection, only to discover that those medications do not work. Then you try other alternatives that can be prescribed to treat those bacteria, only to discover yet again that those medications do not work either. Why? Well, that is likely the result of a methicillin-resistant Staphylococcus aureus, also known as MRSA. What are you supposed to do then? Fortunately, there's a rising solution to that increasingly prevalent and concerning problem. My group, consisting of Josie Marhet Bell, Anthony Fernandez, Tyrone Griffiths, and I, Shayla Melendez, have decided to research the solution in what is known as bacteriophage therapy. Now, to get an understanding of what bacteriophages are, these are viruses which only infect and replicate within bacterial cells. It is interesting to note that phages are extremely diverse as a result of having different sizes, morphology, and genomic material. And just like bacteria, these phages are classified based on their morphology and genomes, separated into families and orders. Most notably, these biological agents are species-specific, meaning that phages are only able to affect one bacterial species. Though, that is not to say they certainly cannot be broad, which is to mean they can infect a particular host range, such as a bacterial genus. Although it is to be noted and pointed out that that is certainly a typical quality of a virus, along with other similar virus-like qualities, such as unable to replicate without a host, using the host structures for their own replication mechanisms, and having small nuclear materials within them. The basic anatomy of a bacteriophage consists of three various forms, such as an icosahedral head and tail combination, just an icosahedral head without having a tail, and a slender thread-like form, also known as a filamentous bacteriophage. Although, at its core, they all contain genetic material within the capsid head. As for the genetic material within the capsid head, bacteriophages can contain either double-stranded or single-stranded DNA or RNA, of which the most common bacteriophage is that of a double-stranded DNA phage with the icosahedral head and tail combination. To go slightly more in-depth, there are two kinds of phages that we are especially familiar with, that being the virulent phage and the temperate phage, which I will further discuss later due to those models both having different kinds of replication strategies, which make them identifiable as a result. Now, due to the overuse and misuse of antibiotics within various industries, such as food and medicine in particular, these biological agents are now seeing a recent revitalization within the medical and science community and research due to their prowess against the war on bacteria, especially that of the rising antimicrobial resistance. That is not to say that they were not being used before, having found proper usage within the food, hygiene, and agricultural industries. These are alternatives as opposed to antibiotics being overused as a result. Just as all viruses do, there are steps to ensure that the virus infiltrates successfully, albeit with different entry mechanisms depending on what or who the virion intends to infect. Such as that, bacteriophages often will puncture through the cell wall using its attached tail tip to directly inject its genome into the cell membrane and cytoplasm of its host cell. That is when the replication mechanisms come into play, with just two strategies known as the lytic and lysogenic cycle, though there are some phages which can carry out both processes. Lytic is where a phage infects a bacterial cell and has its own DNA circularizes, replicating and creating proteins of said phage while remaining apart from the host cell's DNA. Finally, the bacterial cell lyses, once it has completed and utilize all of the host cell's resources, releasing the replicated phages out to continue the cycle once more. As I mentioned earlier, the well-known example of a phage that exhibits this cycle is the virulent T4 phage, which is a double-stranded DNA virus that is part of the Myoviridae family and has a 169 kilobase pair long genome, as shown here in this image. Lysogenic, unlike the lytic cell, does not result immediately in the bacterial cell lysing after replication has been completed. It instead combines the viral DNA with the bacterial host's DNA, replicating with the potential of even becoming a plasmid, which is a circular piece of DNA within the host cell, and existing within the cell until it depletes the resources of the host cell. Once the depletion of the resources has occurred, that is when the phage will then possibly become active, causing the cell to finally lyse. It is important to note that by the time the cell lyses, that the phage has already mixed its own DNA with the host cell's own DNA and has been replicated alongside the daughter host cell. A well-known example of a phage that utilizes the lysogenic cycle, also known as a temperate phage, is that of the lambda phage, which is a double-stranded DNA virus that is part of the Cyphoviridae family and has a 48 kilobase pair long genome, which is also shown here in this example as well. Essentially, the lysogenic infection is within the cell due to the DNA mixture. And the lytic cell is both an infection within and outside because the phage injects its genetic material but does not intermingle it, causing the host cell to burst and spread its replicated phages to other bacterial cells. For efficient utilization of bacteriophage therapy, there are 3 standards of which the phage must pass to be considered, which are as follows. The virus should preferably be from the lytic cycle, be able to effectively kill and thus eliminate the bacterial host cells, and be able to omit side effects. With an understanding of how bacteriophages operate and the criteria that phages must pass in order to be used for therapy, it easily illustrates the capability of using the phages as alternative treatments towards antimicrobial resistant bacteria. As presented by my partners, there are already studies and clinical trials being done in order to test its efficacy.
Hi, my name is Tarini Griffiths, and I am also a part of Group 2. My first article that I will be discussing is titled Successful Treatment of Staphylococcus aureus Prosthetic Joint Infection with Bacteriophage. The main purpose of this experiment was to focus on the safety and efficiency of both intravenous and intraarticular infusions of bacteriophage therapy. The participant in this care was a 61-year-old woman who was successfully treated after a second cycle of bacteriophage therapy administered at the time of a 2-stage exchange procedure for persistent penicillin-sensitive Staphylococcus aureus prosthetic knee joint infection. The patient first underwent bacteriophage susceptibility testing by means of the double agar overlay method or biolog method. This was done with a serial dilution of phage added to a lawn of bacterial isolate grown on an agar plate. The first treatment cycle consisted of a cocktail of 3 phages, which was given 2 hours before an antibiotic dose over a period of 8 weeks. For the second treatment cycle, the patient was given a single lytic phage as well as IV treatment for another 6 weeks. The findings after the first and second course of BT saw no effects. However, phage concentration decreased in the presence of serum obtained on day 1 of treatment. This graph also displays a visual of the patient's results. The results show an incubation using PBS control, pre-treatment serum, and post-treatment serum at 0, 1, 2, and 3 hours. Overall, the graph demonstrates how the concentration of viable phage varies over time. As time goes by, the patient's serum neutralizes the phages, with post-treatment serum being noticeably more effective than pre-treatment serum. The authors noted that failure in the first cycle could be related to the low concentration of phages in the cocktail. However, they were able to demonstrate the safety and ease of administration of BT. The outcome of their experiment was successful as the patient did not have any adverse flare-ups related to either cycle of BT, and the treatment response has been durable for over 20 months with no repetition of MSSA. My second article was titled Highly Different Effects of Phage Therapy and Antibiotic Therapy on Immunological Responses of Chickens Infected with Salmonella, Enterica, Cerovar, Typhimurium. The aim of this study was to determine the chicken immune response to phage therapy by examining the concentrations of cytokines, the percentage of lymphocytes, and the levels of stress hormones. The population in this care were non-genetically modified chickens. The chickens were divided into groups, with one group receiving saline and the other group a phage cocktail over a period of days. The graph shows the effectiveness of phage cocktails and saline on Salmonella typhimurium using 200 chickens, which were randomly divided into 8 groups. The results indicated that the group that received the phage cocktail pro-inflammatory interleukins had significantly increased as opposed to the other control groups after 4 days. It then decreased significantly after 28 days. The results also indicated that phage therapy is effective in removing S. typhimurium from the chicken gut. The authors suggested that phage therapy is effective in the elimination of the pathogenic bacteria and is safe on the immune response of animals. This graph shows how the interferon gamma levels change in chickens in each group after they undergo phage therapy or antibiotic therapy. Terminations 1 to 4 corresponded to days 6, 20, 28, and 24 of the experiment. Overall, phage therapy and antibiotics appear to reduce the immune response to Salmonella infection as indicated by the lower interferon gamma levels when compared to the controls. My third article was titled Bacteriophage Therapy is an Application for Bacterial Infection in China. In this research, they describe the biological characteristics of phage and summarize the phage application in China in mammals, ovipara, aquatilia, and human clinical treatments. In mammals, mice models are often used in the treatment of diseases such as pneumonia, sepsis, and intestinal infection phage therapy, which have been shown to have a good therapeutic effect on them. The diagram shows a summary of the clinical applications of bacteriophage in China and how it is used in various parts of the body. It can be used to treat infections such as lung infection, urinary tract infection, and skin infection. The data collected indicated that phage had a good therapeutic effect on drug-resistant bacteria in veterinary fields, as well as in clinical treatment of humans. The authors suggested that phage therapy is a great way to fight against antibiotic-resistant diseases. However, to achieve this, then we need to take into consideration the lysis spectrum, the immune response, the issue of storage, and the pharmacokinetics of phages. Impacts. The study with the most impacts would be the successful treatment of prosthetic joint infection with bacteriophage. This is because this kind of therapy has potential for treating prosthetic joint infections and is a better alternative than antibiotics for thousands of patients. The results from the study by Sanchez-Gonzalez et al. Suggest that bacteriophage therapy is effective in eradicating infection strains in prosthetic joint infections and is better tolerated and has fewer side effects. Prosthetic joint infection cases can help bacteriophage therapy in that this kind of practice can become more widespread worldwide. It can also help in outlining some of the challenges of phage therapy, such as routes of administration, dosages, and duration of therapy. Also, it can help researchers to figure out if phage therapy can replace antibiotics and to broaden our understanding of the value of phage therapy in clinical practice.
Hello, everyone. My name is Josie Mary Bell, and I will continue on with the drawbacks. Bacteriophage therapy have a lot of drawbacks, and one of the key drawbacks of bacteriophage therapy is its limited host range. Phages have limited host range, and they target specific bacteria, not mixed infections. Unlike antibiotics, which often have broad spectrum activity, bacteriophages are highly specific, and they only target certain bacterial strain based on unique markers on the bacterial surface, which specifically could be both an advantage and a limitation. While it reduces collateral damage to beneficial bacteria, it also means that these phages are ineffective against bacterial strains without the matching receptors. And to better explain this, let's focus here on the image to the right, to the image on the right. At the top, you see a bacteriophage, which is a virus that infects the bacteria. And the bacteriophage uses its tail fibers, which are those tiny leg-like structures, to attach to specific surface receptors on the bacterial cell. The bacterial surface has a variety of receptors, and each one of them corresponding to different molecular structures. These would include glycolipids, which as we all know, are complex sugar-like molecules found in bacterial membrane. You have OMPF, which is an outer membrane protein involved in transporting small molecules. You have B-tube, which is a transporter protein that brings in vitamin B12. And over here, we have the Tol-C, which is a channel protein linked to bacterial efflux system, which the bacteria uses to expel any harmful substances. And lastly here, we have the flagellar proteins, which are, of course, found on structures that help bacteria move. These arrows right here in the image actually indicate how a phage matches with a specific receptor. And if the bacteria lack these surface markers, the phage cannot attach and therefore cannot infect the bacteria, which makes sense because in order to use phage therapy, doctors must first identify the exact bacterial strain causing the infection and ensure that it matches a known phage. And if the strain is misidentified or perhaps the phage doesn't match, what's going to happen is the therapy wouldn't work. This process could take a lot of time, which is a serious limitation when treating life-threatening infections. And this only means that a phage that is effective against one bacterial strain may not always work against another, delaying treatment if that exact strain is not identified. Now, moving along, another drawback is the immune interference, which if you see in the picture here, you have the little body, and then you have the phages that are coming and how it's interfering against it. So the body may neutralize phages before they even work. And yes, the immune response can diminish phage effectiveness. So with these, the phages are naturally recognized by the immune system as foreign invaders, and the immune system can launch an attack to neutralize those phages, which reduces their effectiveness. And it's like when your body fights off a virus or an infection. The same way, the phages, despite their potential benefits, can be destroyed by our body's immune response before they even have a chance to do their job. Now, moving on to storage issues, which is next slide over here. Phages need precise conditions to remain to remain viable. And bacteria can adapt to evade those phages. Indeed, phages require specific conditions to remain in order for them to remain stable and effective. And if they are not stored at the correct temperature, they can quickly lose their ability to target bacteria. This means that in low resource settings or in areas with unreliable refrigeration, these phage therapy could become impractical, and storage challenges also limit phage availability, especially in low resource settings. Another drawback, bacteria can evolve to resist phages through a mechanism like CRISPR, Cas system, or receptor modifications. And just like bacteria can develop resistance to antibiotics, they can also develop resistance to bacteriophages. And over time, the bacteria may actually evolve mechanisms to evade phage attacks, making the treatment less effective. And phage-resistant strains could emerge, which is a major concern for the long-term sustainability of phage therapy. Last but not least on our list, we have dosages and delivery methods require further optimization. Basically, the way phages are delivered, how much is issued, and how long the treatment lasts, these are still under study. And for example, the study on prosthetic joint infections did highlight how difficult it is to actually figure out the right dosages and the best way to deliver these phages to the infection site. But seems to be our time. I'm going to stop here on the drawbacks in order to carry on with the future implications.
Okay, so for the conclusion and future implications, firstly, good morning or good afternoon, everyone. My name is Anthony Fernandez, and today I will be concluding our presentation by summarizing the insights shared by my team and discussing the conclusion and future implications of bacteriophage therapy. As the final speaker, I have the privilege of tying everything together that was already said and highlighting why this therapy is so significant. Let me start by summarizing the major findings of our presentation. In the first article, we heard about the successful treatment of a prosthetic joint infection caused by Staphylococcus aureus. This case involved a 61-year-old woman who had a challenging infection in her prosthetic knee joint. The patient received 2 cycles of bacteriophage therapy, one with a cocktail of different phages and another with a single lytic phage in combination with already existing antibiotics. Although the first cycle faced challenges, adjustments in things like the phage concentration and the administration allowed for a long-term positive outcome. The infection in this case did not recur for over 20 months, showcasing the safety, durability, and customizability of bacteriophage therapy. Next, in the second article, we learned about a study on phage therapy in chickens infected with Salmonella enterica. This research demonstrated that bacteriophage therapy effectively eliminated the infection while also influencing immune responses. Specifically, the therapy caused a temporary spike in inflammatory markers, which later stabilized. These findings highlight the potential for phage therapy to not only treat infections, but also modulate immune responses, an essential consideration for its application in humans. Finally, in the last article, we explored the application of bacteriophage therapy in China. The research presented included treatments for diseases in mammals, aquatic animals, and even humans. From combating pneumonia and sepsis in mice to addressing antibiotic-resistant bacteria in humans, the studies underscore the versatility of phage therapy. However, there were issues in this study, challenges like storage stability, the variability of phage activity, and pharmacokinetics remain areas for improvement for future studies. Bringing all these findings together, we can draw several conclusions. Bacteriophage therapy is more than just an alternative to antibiotics. It represents a transformative approach to addressing drug-resistant infections. Its adaptability and precision, as shown in both the animal and human studies, make it a promising tool for the future for healthcare providers. Additionally, the therapy's fewer side effects when compared to conventional antibiotics enhance its appeal to the general population as a safer treatment option. Looking ahead, the future of bacteriophage therapy holds great promise. First, continued research and development will be critical to refining delivery methods, optimizing dosages, and exploring new combinations of phages. Overcoming these challenges will pave the way for more widespread and effective use of this therapy. Second, bacteriophage therapy could play a major role in combating antibiotic-resistant infections. As bacteria will only continue to evolve over time and evolve to be more resistant to already existing treatments, phages offer a highly targeted solution that could save countless lives. Third, there's also applications beyond medicine. Bacteriophages can also find applications in food safety, environmental sanitation, and even industrial cleaning. You can picture a world where phages are commonly used to reduce harmful bacteria in things like food production facilities or even to purify water samples. Along with this, we must also consider the potential for integrating this bacteriophage therapy into routine healthcare globally. In the future, doctors might have access to phage banks, which will allow them to select specific phages depending on their patient's infection. This precision medicine approach could revolutionize how we treat bacterial diseases. At the same time, global collaboration is necessary and will be essential to establish regulatory frameworks and ensure equitable access to this therapy worldwide. While the potential for this therapy is immense, there are challenges that inevitably have to be addressed. That brings up the complications. Each bacteriophage is very unique, so standardizing their production and their quality across different settings is complex. Clear regulatory guidelines for approval and monitoring will also be needed to ensure safety and consistency, no matter where it's found in the world. Additionally, making phage therapy affordable and accessible, especially in resource-limited settings such as in impoverished country, will require partnerships between many different moving parts, such as the government or research centers or even private companies to help distribute. In conclusion, bacteriophage therapy is not just a solution for today's challenges, but a path towards a healthier future. It addresses the urgent issue of antibiotic resistance while opening new doors in medical and environmental applications. By investing in research, addressing logistical challenges, and educating the public, we can make this innovative therapy a mainstream option when compared to just normal antibiotics. Thank you very much for your attention. I look forward to any questions or thoughts on this exciting topic. These are our resources for the photos used in the PowerPoint and icons, and these are our references. Thank you very much.